The coldest marine environment on Earth is not a place that invites life. It is a vast expanse of ice and dark water, where temperatures rarely rise above freezing and immense sheets of ice stretch across the horizon as far as the eye can see. The Antarctic Ocean, with its harsh conditions and unforgiving cold, seems almost inhospitable. One might expect that only the occasional penguin, seal, or hardy crustacean could endure such extremes. Yet life persists, often in forms that defy expectation. Among these forms, there exists a group of creatures that has evolved in ways so extraordinary. It challenges fundamental assumptions about how vertebrates function. They are the ice fish, and their biology is unlike anything found elsewhere in the animal kingdom. These fish inhabit waters that hover just below 0 degrees Celsius for much of the year. In summer, the ocean might reach a fleeting 2 degrees Celsius, but in winter, it sinks to nearly minus 1.8 degrees. Despite the apparent scarcity of resources and the frigid conditions, these waters are rich in oxygen, more so than tropical or temperate oceans. The solubility of gases increases as temperatures fall, meaning that, counterintuitively, the Antarctic Ocean offers more dissolved oxygen than warmer seas. For most animals, this alone would not be enough to overcome the challenges posed by such extreme cold. For ice fish, however, the abundance of dissolved oxygen becomes an unexpected avenue through which evolution reshapes the very foundation of life. What sets ice fish apart is immediately apparent upon closer examination. Their blood is almost entirely transparent. This is not a subtle tint or a faintly unusual shade. It is a nearly complete absence of color, an anomaly among vertebrates. Hemoglobin, the protein responsible for transporting oxygen in red blood cells, is missing. Hemoglobin is fundamental to vertebrate life. In almost every other fish, in birds, in mammals, in reptiles, and amphibians, it is the central mechanism by which oxygen moves from the respiratory organs to tissues throughout the body. It carries life-sustaining oxygen to muscles and organs, and without it, cells cannot function. They suffocate, metabolism grinds to a halt, and death follows rapidly. For ice fish, the absence of hemoglobin is more than a curiosity. It is a radical departure from what we understand as the baseline for vertebrate life. Even more remarkable is that these creatures also lack myoglobin in their muscles, the protein that stores oxygen within muscle tissue. Myoglobin is closely related to hemoglobin in structure and function, but it operates at the cellular level, ensuring that muscles have immediate access to oxygen during activity. In most animals, both hemoglobin and myoglobin work in concert, creating a highly efficient system for oxygen transport and storage. Ice fish, however, operate without either. As a result, their muscles and even some organs appear almost completely transparent, a ghostly glimpse into their internal anatomy. A few species retain minute amounts of myoglobin in the heart, but this is the exception rather than the rule. Genetic studies have revealed a further layer of mystery. These fish still carry the genes for both hemoglobin and myoglobin, but these genes have been switched off over the course of evolutionary history, rendering them non-functional. The reason for this switch is a story of environmental pressures, genetic chance, and adaptation to one of the most extreme habitats on Earth. The Antarctic Ocean itself is a place of extremes. Its waters are cold enough to threaten freezing, yet the richness of dissolved oxygen provides a resource that most other ecosystems lack. There are relatively few species of fish here, only about 374 recorded so far, a stark contrast to warmer and more diverse ecosystems such as the Great Barrier Reef, which supports many times more species in a fraction of the area. The scarcity of life is offset by the uniqueness of what survives. Ice fish, like other Antarctic inhabitants, are extremophiles. Every aspect of their physiology is shaped by the demands of survival in near-freezing waters. and their transparent blood is only the most visible of these adaptations. Evolution has reshaped them at a molecular, cellular, and systemic level, forcing them to find solutions to problems that would be lethal to almost any other vertebrate. The lack of hemoglobin, in particular, is astonishing. Hemoglobin is an iron-containing protein that not only gives blood its familiar red color, but also has an extraordinary chemical capacity to bind oxygen and carbon dioxide. In humans and most other animals, red blood cells packed with hemoglobin pick up oxygen in the lungs or gills, carry it through the circulatory system, and deliver it to organs and muscles where it is needed most. Carbon dioxide, a metabolic waste product, is transported in the reverse direction for elimination. This system is highly efficient, allowing animals to survive under conditions of high activity, fluctuating oxygen availability, and metabolic stress. Some invertebrates, such as octopuses or certain mollusks, utilize a different protein, hemocyanin, which contains copper instead of iron and lends their blood a blue-green hue. Hemocyanin functions similarly, transporting oxygen and carbon dioxide, although with different efficiency and under different environmental constraints. Icefish, however, dispense entirely with oxygen-binding proteins, relying instead on a different set of adaptations to survive. Without hemoglobin or myoglobin, icefish face an obvious problem: how to deliver sufficient oxygen to sustain cellular function. The answer lies in a combination of environmental factors and evolutionary compensations. Cold water, rich in dissolved oxygen, means that the oxygen content of the environment is unusually high. Icefish gills have evolved to be larger than those of most other fish, maximizing the surface area for oxygen extraction. Their skin, unusually smooth and devoid of scales, also participates in oxygen absorption. This cutaneous respiration, common in amphibians but rare in fish, supplements gill-based intake and ensures that oxygen can diffuse directly into the bloodstream. In effect, icefish absorb dissolved oxygen directly into plasma, the liquid component of blood. The process is far less efficient than hemoglobin-mediated transport, but in oxygen-saturated waters, it is sufficient to support this inefficient circulatory system. Icefish have undergone remarkable anatomical changes. Their blood is far less viscous than that of red-blooded vertebrates, a property that allows it to flow more easily through vessels at lower pressures. To further compensate, they have larger blood volumes and expanded blood vessels, increasing the overall capacity to transport oxygen despite the absence of hemoglobin. Their hearts are enormous relative to their body size, in some species up to 5 times heavier than those of closely related red-blooded fish. This enlarged heart pumps large volumes of blood with each contraction, creating a high cardiac output that circulates oxygen-rich plasma throughout the body. These adaptations demonstrate the intricate ways evolution can compensate for a trait that would otherwise be catastrophic. Metabolic adaptations complement these anatomical changes. Icefish have extremely slow metabolic rates, often estimated to be as much as 75% lower than those of similar-sized fish in warmer waters. This low metabolic demand reduces their need for oxygen, making survival without hemoglobin more feasible. Activity levels are similarly restrained. Most icefish spend long periods motionless or move slowly along the seafloor. They are ambush predators, lying in wait for unsuspecting prey to approach rather than actively pursuing it. This strategy conserves energy and minimizes oxygen requirements, a necessary adjustment for creatures with such an unconventional circulatory system. The evolutionary story of icefish is both extraordinary and instructive. The complete loss of hemoglobin and myoglobin may seem maladaptive, given how essential these proteins are to other vertebrates. One hypothesis is that a genetic mutation disabling hemoglobin production arose in an ancestral population. In small, isolated populations where mating choices were limited, this mutation persisted despite its potential cost. The Antarctic environment, with abundant dissolved oxygen and limited competition, allowed survival while alternative physiological adaptations evolved. Over time, these compensatory mechanisms enlarged hearts, expanded blood volume, cutaneous respiration. Slow metabolism became fixed in the population, illustrating how evolution can accommodate even seemingly disadvantageous changes under the right conditions. Studying icefish also provides insights that extend far beyond the Antarctic. Understanding how vertebrates can survive with minimal oxygen transport has implications for human medicine. The unique properties of icefish blood, including its low viscosity and high plasma oxygen solubility, could inform the development of artificial blood substitutes or treatments for circulatory disorders. Their adaptations offer a living laboratory for exploring how hearts, vessels, and metabolism can be modified to function under extreme constraints. In the long term, these insights may even inform approaches to extreme environments, whether the deep ocean or space, where oxygen availability is limited and efficiency is paramount. The icefish reminds us that biology is not a static set of rules, but a dynamic process shaped by environment, chance, and necessity. Traits that appear essential in one context can be discarded in another when circumstances allow. Life in the Antarctic Ocean has created a vertebrate unlike any other, one that challenges our understanding of circulation, metabolism, and survival. By examining the icefish, we gain not only a window into evolutionary creativity, but also a glimpse of the possibilities that emerge when the constraints of the familiar are lifted. In a place where the water is perpetually near freezing and oxygen is abundant, evolution has redefined what it means to be a vertebrate, producing a creature that survives without the very proteins we assume are indispensable. The icefish is a testament to the resilience and adaptability of life, a reminder that even in the most extreme conditions, nature finds a way. Despite the absence of hemoglobin and myoglobin, icefish thrive in an environment that would be lethal to most other vertebrates. Their biology, at first glance, seems almost contradictory. How can a vertebrate survive without the very proteins considered essential for life? The answer lies in a combination of environmental context, physiological specialization, and evolutionary contingency. Icefish exist in a world of extremes, where low temperatures, high oxygen availability, and geographic isolation intersect to create conditions that permit extraordinary adaptations. Their survival strategy is a delicate balance, a combination of physical, molecular, and behavioral modifications honed over millions of years of evolutionary experimentation. The circulatory system of icefish is fundamentally different from that of most vertebrates. The absence of hemoglobin reduces the oxygen-carrying capacity of the blood by over 90%. In response, icefish blood has become almost entirely plasma, with dissolved oxygen moving freely throughout the body. The blood itself is less viscous than that of other fish, meaning it flows more easily, even at the low pressures typical of Antarctic waters. This low viscosity reduces the energy required to pump blood, an important adaptation in creatures whose hearts are already burdened with the task of moving oxygen-poor plasma. In practical terms, the blood of an icefish flows rapidly through its large vessels, ensuring that oxygen can reach tissues efficiently despite the lack of specialized transport proteins. These large vessels are a critical adaptation. Arteries and veins in icefish are wider than in related species, allowing more blood to circulate at any given time. The total blood volume is also significantly greater, up to 4 times that of other Antarctic fish of similar size. This increased capacity ensures that, at any given moment, more oxygen is available for diffusion into cells. Even with this adaptation, oxygen transport is inherently less efficient than in red-blooded fish. To compensate, the cardiovascular system has evolved to maximize throughput. The hearts of ice fish are disproportionately large, with some species exhibiting hearts that are up to 5 times heavier than those of similar-sized red-blooded relatives. Each contraction of such a heart moves a massive volume of blood, maintaining a circulation rate sufficient to sustain life. The heart of an ice fish is not only large, but also powerful in its output. This high cardiac output is necessary because dissolved oxygen in plasma alone is far less efficient at delivering oxygen than hemoglobin-bound oxygen. In addition to the increased heart size, ice fish hearts exhibit structural specializations, including expanded chambers and thickened walls, allowing them to handle greater blood volumes without excessive strain. Unlike active predators such as cheetahs, which have large hearts to support bursts of speed, the oversized heart of the ice fish serves an entirely different purpose, maintaining oxygen delivery in a system where oxygen transport is inherently limited. The heart's function is less about meeting sudden high metabolic demands and more about sustaining life in an environment where energy conservation is paramount. Metabolism is another crucial component of ice fish survival. Living in near-freezing waters reduces the need for rapid energy expenditure, and these fish have correspondingly slow metabolic rates. Estimates suggest that their standard metabolic rate is up to 75% lower than that of similarly sized fish in temperate waters. This low metabolism is a key compensatory mechanism, minimizing the amount of oxygen required for basic cellular function. Slow movement and sedentary behaviors further reduce oxygen demand. Ice fish are ambush predators, lying in wait for prey rather than pursuing it over long distances. This strategy conserves energy, aligning with their physiological limitations, and allows them to remain viable despite a circulatory system that would be considered inefficient under most circumstances. Oxygen uptake in ice fish is a combination of gill and skin absorption. Their gills are unusually large, with increased surface area to maximize oxygen extraction from water. This is particularly important because the oxygen must dissolve directly into plasma rather than bind to hemoglobin. In addition, their scales are absent, leaving smooth, permeable skin capable of cutaneous respiration. Oxygen diffuses directly through the skin into the bloodstream, a method common in amphibians but rare in fish. While this process is less efficient than hemoglobin-mediated transport, it is sufficient in the oxygen-rich waters of the Antarctic Ocean. Together, these adaptations allow ice fish to survive and even thrive despite the absence of oxygen-binding proteins. The genetic story behind ice fish is equally remarkable. They retain the genes necessary to produce hemoglobin and myoglobin, yet these genes are silenced. The mechanisms behind this silencing are a subject of ongoing research, but it appears that regulatory mutations prevent expression of these essential proteins. In other words, ice fish could produce hemoglobin and myoglobin, but evolution has switched off the ability to do so. This selective silencing represents a profound evolutionary event, suggesting that in certain ecological contexts, traits once considered indispensable can become redundant or even disadvantageous. The reasons for this shift are likely multifactorial, involving both genetic drift and selective pressures imposed by extreme environmental conditions. One hypothesis is that a mutation disabling hemoglobin production occurred in an ancestral population of ice fish. In small, isolated populations, common in the harsh Antarctic environment, genetic variation is limited and selection pressures are constrained. Individuals with the mutation may have survived simply because environmental conditions allowed it, and without competition from closely related species, the trait persisted. Over generations, compensatory adaptations, including enlarged hearts, Increased blood volume and enhanced gill function became fixed in the population, allowing the species to flourish despite the absence of hemoglobin. This scenario illustrates how evolutionary outcomes are often contingent on context, with chance mutations interacting with environmental conditions to produce outcomes that might seem counterintuitive or maladaptive in other settings. The evolution of icefish also underscores the importance of energy conservation in extreme environments. Producing hemoglobin and myoglobin requires significant metabolic investment, both in terms of protein synthesis and maintenance of red blood cells. In an environment where oxygen is abundant but temperatures are low and metabolic rates must remain minimal, eliminating the production of these proteins may have offered a subtle energetic advantage. By dispensing with hemoglobin and myoglobin, icefish reduce the energetic cost of maintaining a circulatory system, reallocating resources toward other adaptations necessary for survival. The result is a highly specialized organism, optimized for a narrow but stable ecological niche. The peculiar biology of icefish extends beyond oxygen transport. Their transparent bodies, resulting from the absence of myoglobin and muscles, create a ghostly appearance that has fascinated researchers for decades. Some species appear almost entirely see-through, with only the heart and internal organs faintly visible beneath the skin. This transparency is not merely an aesthetic quirk. It reflects a fundamental rearrangement of internal physiology, a manifestation of how evolution can modify the structure of life at multiple scales. It also raises intriguing questions about the limits of vertebrate design. If a vertebrate can function without hemoglobin and myoglobin, what other assumptions about essential biological systems might be challenged? The implications of studying icefish extend into medical research. Their low viscosity, oxygen-rich plasma offers a natural model for understanding fluid dynamics in the circulatory system. Insights from icefish blood could inform the development of artificial blood substitutes, treatments for cardiovascular disease, and strategies for improving oxygen delivery in critical care settings. Researchers are investigating how plasma alone can carry sufficient oxygen to sustain life, a concept that could lead to innovations in trauma medicine, organ preservation, and even long-term spaceflight. By observing how evolution has solved the problem of oxygen transport without hemoglobin, scientists gain a living blueprint for alternatives to traditional vertebrate physiology. Beyond medicine, the study of icefish has broader ecological and evolutionary significance. These fish are a striking example of how life can adapt to extreme conditions, providing insight into the potential for resilience and flexibility in biological systems. They demonstrate that evolution does not follow a rigid path, but is instead responsive to environmental context, available resources, and stochastic events such as genetic mutation. The story of the icefish highlights the interplay between constraint and opportunity in evolutionary history. It shows that traits considered universal, such as the presence of hemoglobin, may be dispensable under specific ecological conditions, revealing a plasticity in vertebrate design that is both surprising and instructive. Icefish also provide a lens through which to examine broader questions about the limits of life. How far can physiology be pushed before survival becomes impossible? What trade-offs are acceptable in extreme environments? In the case of icefish, oxygen transport efficiency is sacrificed. Metabolic rates are reduced and behavioral strategies are constrained. Yet survival is not only possible but sustainable over evolutionary timescales. These trade-offs illustrate a principle that extends across biology. Adaptations are rarely perfect, and evolution often works by balancing competing demands, producing organisms that are optimized for specific environments rather than universally superior. The history of ice. Fish evolution is intertwined with the geophysical history of the Antarctic region. As the continent became isolated and glaciation intensified, cold-adapted lineages diverged from their temperate relatives. Geographic isolation, limited competition, and selective pressures unique to polar ecosystems created conditions in which unusual traits could persist and proliferate. The combination of high oxygen solubility in cold water, slow metabolism, and energetic constraints created an environment in which the loss of hemoglobin and myoglobin became viable. Over millions of years, these adaptations stabilized, resulting in the remarkable species we observe today. The icefish is thus both a product of its environment and a testament to the power of natural selection. Operating under extreme conditions, behaviorally, icefish have also adapted to their frigid habitat in subtle but important ways. Ambush predation minimizes energy expenditure, while slow, deliberate movements reduce oxygen demand. Their feeding strategies, largely opportunistic, reflect the scarcity of prey in Antarctic waters. Reproductive behaviors are similarly constrained, with low-density populations and limited mobility influencing mating patterns and gene flow. These ecological pressures, combined with physiological specializations, create a coherent survival strategy in which every aspect of the organism is finely tuned to its environment. The icefish exemplifies a form of life that is integrated, efficient, and in its own way, elegant. From a broader perspective, the icefish challenges fundamental assumptions about vertebrate physiology. It demonstrates that traits long considered indispensable may be bypassed under the right conditions. It raises questions about the evolutionary plasticity of life, the interplay between environmental constraints and genetic variation, and the myriad ways in which organisms can adapt to seemingly inhospitable habitats. For scientists, these fish are more than curiosities. They are models for exploring the limits of adaptation, the mechanics of oxygen transport, and the potential for novel solutions to physiological challenges. In exploring the Antarctic Ocean and the biology of icefish, we are reminded of the astonishing capacity of life to persist against odds. These creatures inhabit a world of ice and near-freezing water, yet they have evolved solutions that allow them to survive without the very proteins we associate with life itself. They absorb oxygen through their skin and gills, circulate plasma with unusually low viscosity, maintain enlarged hearts and blood vessels, and rely on slow metabolism and energy-conserving behaviors to thrive. In doing so, they challenge our understanding of biology, offering lessons that extend from the molecular to the ecological and even into the realm of human medicine and technological innovation. Icefish exist at the intersection of extremes, a convergence of environment, genetics, and evolutionary history that produces a creature unlike any other vertebrate on Earth. Their story begins not just with the biology of individual fish, but with the larger forces that have shaped the Antarctic Ocean itself. For millions of years, this southernmost ocean has been isolated from the warmer waters of the world. Its temperatures stabilized near the freezing point, its surface locked beneath thick ice sheets. These conditions have acted as both a barrier and a crucible, limiting competition, reducing predation pressures, and allowing unique evolutionary experiments to unfold. In such isolation, small populations of fish were able to survive and evolve in ways that would be improbable and more competitive. Diverse environments. The silencing of hemoglobin and myoglobin in icefish represents one of these evolutionary experiments. Genetic analyses suggest that ancestral populations experienced mutations that inactivated the production of these proteins. In most circumstances, such mutations would be catastrophic, leading to rapid mortality. Yet, in the oxygen-rich, frigid waters of the Antarctic, the selective pressures that normally eliminate such traits were relaxed. Small, isolated populations amplified the effect of genetic drift, allowing this unusual trait to persist. Over generations, compensatory adaptations emerged: larger gills, smooth and permeable skin, enlarged hearts, expanded blood vessels, increased blood volumes, and slow metabolic rates. Each adaptation interacted with others in a complex network, stabilizing survival in an environment where conventional vertebrate physiology might fail. The evolutionary implications of ice fish are profound. Their existence demonstrates that traits once considered indispensable are not immutable. They can be lost or altered if environmental conditions allow. Life is not constrained by rigid formulas, but is instead responsive, flexible, and experimental. Evolution works not only by enhancing advantageous traits, but also by accommodating the survival of organisms with seemingly disadvantageous characteristics, provided that other compensatory mechanisms can mitigate the consequences. The icefish is a living example of how natural selection operates in conjunction with chance, geography, and environmental context to produce forms of life that challenge preconceived notions of what is necessary for vertebrate survival. This adaptability extends beyond the molecular and anatomical to behavior and ecology. Icefish are ambush predators, relying on patience and strategic positioning rather than speed and endurance. Their feeding behaviors are opportunistic, reflecting the scarcity of prey in Antarctic waters, while their reproductive strategies are influenced by low population densities and limited mobility. Every aspect of their life is tuned to conserve energy and optimize oxygen utilization. Their slow movements, extended periods of stillness, and carefully timed predatory actions illustrate a broader principle. In extreme environments, survival often favors efficiency and restraint over rapid activity and metabolic expenditure. Studying icefish provides insight not only into evolution but also into fundamental principles of physiology. Their circulatory system, reliant on plasma rather than hemoglobin, challenges our understanding of oxygen transport and raises questions about the limits of biological design. Researchers examining icefish blood are exploring its low viscosity, high oxygen solubility, and efficient diffusion as models for human medical applications. Understanding how these fish circulate oxygen without hemoglobin could inspire artificial blood substitutes, new strategies for managing cardiovascular disease, and innovative approaches to trauma care. The lessons from icefish extend even further, suggesting possibilities for human survival in extreme or low oxygen environments. From deep-sea habitats to outer space, the biology of icefish also invites reflection on the boundaries of adaptation. In a world where oxygen is abundant but temperatures are lethal to most life, these fish have discarded traits that we consider fundamental, yet they survive. This suggests that the limits of vertebrate physiology are more flexible than previously assumed, contingent on environmental context and the presence of complementary adaptations. If a vertebrate can function without hemoglobin in one of the harshest environments on Earth, it raises intriguing questions about what is possible elsewhere. Could similar principles allow other vertebrates to adapt to extreme or novel environments? How might human physiology be engineered or assisted to cope with conditions that currently seem beyond our biological limits? Icefish serve as a model, demonstrating that adaptation is not just a story of incremental improvements, but can also involve radical restructuring of what we consider essential. The transparency of icefish, resulting from the absence of myoglobin, provides an additional layer of insight. Their ghostly bodies reveal an anatomy that is usually hidden, offering a unique perspective on the interplay between form and function. Muscles, organs, and internal structures can be observed in a way that is almost entirely invisible in red-blooded vertebrates. This transparency is not merely an aesthetic or scientific curiosity. It reflects a fundamental evolutionary compromise, a visible manifestation of physiological trade-offs that have been carefully balanced to ensure survival. Life, in the case of the ice fish, is a negotiation between structural efficiency, functional necessity, and environmental constraint. The lessons of ice fish extend to considerations of energy use and survival strategy. In the frigid Antarctic, energy is scarce and metabolic rates are low. Movement is slow, predation is patient, and activity is minimized. The energy saved by not producing hemoglobin and myoglobin is invested in structural and circulatory adaptations that maximize survival. This delicate balance underscores a broader principle of biology. Adaptation is about optimization, not perfection. Organisms do not evolve to achieve an ideal form. They evolve to survive and reproduce within the context of their environment. The icefish exemplifies this principle, demonstrating that survival may require discarding traits that seem fundamental, provided that other compensatory mechanisms can maintain function. Icefish also highlight the interplay between genetic potential and environmental reality. Though they possess the genes for hemoglobin and myoglobin, these genes are silenced, their expression blocked by evolutionary processes. The potential exists, but it is not realized. This disconnect between genetic capacity and phenotype illustrates the complex pathways through which evolution operates, showing that survival is not determined solely by what an organism could theoretically do, but by what it actually does in response to its environment. In this way, the icefish serves as both a case study in evolutionary biology and a window into the mechanisms of gene regulation, adaptation, and survival. From a broader scientific perspective, icefish challenge assumptions about oxygen dependency and vertebrate design. They reveal that life can tolerate radical deviations from the norms observed in most animals. By functioning without hemoglobin, they provide a natural experiment in circulatory efficiency, oxygen diffusion, and metabolic regulation. Their existence suggests that alternative pathways to survival are possible, and that the constraints we often assume are absolute may, in fact, be flexible under the right environmental and evolutionary conditions. Icefish are, in essence, proof that biology is inventive, capable of solutions that exceed the boundaries of human expectation. The study of these fish also has implications for understanding climate change and environmental adaptation. As the Antarctic Ocean experiences shifts in temperature, icefish populations may provide insights into how species respond to changing conditions, both physiologically and behaviorally. Their specialized adaptations make them vulnerable in some respects, yet their evolutionary history demonstrates resilience and capacity for coping with environmental extremes. Observing how icefish interact with their changing habitat can inform broader questions about ecological plasticity, species survival, and the potential for adaptation in the face of global environmental change. In reflecting on the icefish, one is struck by the elegance and strangeness of evolutionary innovation. These creatures are simultaneously fragile and robust, highly specialized yet flexible, dependent on extreme conditions yet capable of thriving within them. They force a reconsideration of what is essential in vertebrate biology, showing that even traits considered fundamental can be bypassed, altered, or eliminated if circumstances allow. Their story is a reminder that life is not constrained by human expectations, that evolution is both a sculptor and a tinkerer, shaping organisms in response to pressures that are sometimes subtle, sometimes brutal, and sometimes entirely unpredictable. Ultimately, icefish teach us about the possibilities of life at the edge. They demonstrate that survival does not always require adherence to established rules, and that adaptations can be both radical and effective. They challenge our understanding of oxygen transport, circulatory physiology, metabolic regulation, and evolutionary strategy. They inspire questions about the potential for life in extreme environments, both on Earth and beyond. In their translucent bodies and unconventional biology, they carry the story of evolution's ingenuity, a narrative that is both scientifically rich and profoundly humbling as we explore the biology of icefish. We are confronted with the delicate balance of life, the interplay of environment and evolution, and the extraordinary lengths to which organisms will go to survive. These fish, living in waters that remain perpetually near freezing, reveal the capacity of vertebrates to function in ways that once seemed impossible. They absorb oxygen without hemoglobin, circulate plasma through oversized hearts and vessels, maintain low metabolic rates, and conserve energy through patient ambush predation. They are a testament to the resilience of life and a reminder that the natural world often defies expectation. The icefish exists at the edge of possibility, a living example of the creative power of evolution and the profound adaptability of life in even the harshest conditions. In studying these remarkable creatures, we gain more than an understanding of a single species. We gain insight into the mechanisms of adaptation, the limits of vertebrate physiology, and the potential for biology to solve problems that, from a human perspective, appear intractable. Icefish invite us to reconsider what is possible, to explore how life can thrive without what we consider essential, and to imagine the hidden ingenuity that may exist in other corners of the natural world. They are both a scientific marvel and a symbol of life's resilience, a testament to the power of evolution to reshape the very foundations of existence, even in the coldest and most forbidding environments on Earth.